Hello everyone, this is Dr. Vishal Trivedi from Department of Biosciences and Bioengineering, IIT Guwahati. And what we were discussing about the biomolecules. And in the previous module, we have discussed about the three biomolecules. We have discussed about the nucleic acids, and we have discussed about the carbohydrates, and we have also discussed about the lipids. And in the previous lecture, we have discussed about the proteins. So we have started discussing about the proteins, that the proteins are made up of the amino acids, and these amino acids are having a general structures where they have the central C alpha carbon, and on this central C alpha carbon we have the four different types of functional groups which are attached to each. On one side it has the amino group, on the other side it has the carboxyl group, and the third side it has the hydrogen, and the fourth side it has the functional side chains. And based on the functional side chains, the amino acid can be classified into four different groups, 20 different types. It could be vary from the very simple glycine to a very complicated tryptophan, and depending on the side chain it can be of the different molecular weights and different types of properties. So it could be a hydrophobic amino acids, it could be a hydrophilic amino acids, it could be a polar amino acids, it could be negatively charged amino acid, or it could be a positively charged amino acid. And in addition to that, we have also discussed in detail about the thin layer chromatography and how the thin layer chromatography can be used to analyze the different types of amino acids. So in today's lecture, we are going to discuss more about the protein structures. So let's start discussing about the protein structures. So as we said, protein is made up of the 20 naturally occurring amino acids. A typical amino acid contain a amino group and a carboxyl group attached to the central alpha carbon. The side chain attached to the central alpha carbon determine the chemical nature of the different amino acids. So what you see here is you have the C alpha carbon, and that C alpha carbon is attached to the four different types of functional groups, and these four different types of functional groups, and depending on the R side chains you can have the 20 different types of amino acids. Peptide bond is for connecting the two individual amino acids, and that's how they are actually giving the polypeptide chain. Each amino acid is linked to the neighboring amino acid through an acid amide bond between the carboxyl group and the amino group of the next amino acids. Every polypeptide chain has the free amino group and the free C-terminal groups, right? That's why the primary structure of a protein is defined as the amino acid sequence from the N to the C terminals with a length of the several hundred amino acids. So these are the 20 different amino acids what we have already discussed in the previous lectures. So the primary structure is defined as the amino acid sequence from the N terminus to the C terminus, right? So do you see that it starts with the N terminus? So this is the N terminus, and then it ends up with making a combination with all other amino acid to the peptide bond, but ultimately with the last amino acid it is going to have the carboxyl group which is going to be free. Then that's why the protein is going to have the amino group, and then it is going to have all the amino acids, and then it is going to have the carboxyl group. So that's why the protein has the two ends, one is N terminus and the other one is called as the C terminus. The ordered folding of the polypeptide chain give rise to the 3D conformation known as the secondary structure of the protein, such as the helix, sheets and loops. So this is what you see here is the primary structures, right? So all the protein, all the amino acids are present, and the first amino acid is actually going to have the N terminus, whereas the last amino acid is going to have the C terminus. When this primary structure is getting folded, it is actually going to give you the secondary structures where you have the alpha helices. These are the alpha helices. Then we have the beta sheets. So you can see the beta sheets. These are the beta sheets, and then it also has the turns. So these are the turns what you see here. Arrangement of the secondary structure give rise to the tertiary structures. Alpha helix and beta sheets are connected by the unstructured loop to arrange themselves in the protein structure, and it allows the secondary structure to change their directions. Tertiary structure define the structure of a protein and the enzymatic activity or the nature of the structural protein. So, once the secondary structures are joined together by the loops or the turn, they are actually going to give you the tertiary structure. So, this is what you see here is the tertiary structures, and if the protein has the multiple subunits, then it is actually going to give you the quaternary structure. For example, in this case we have the subunit 1, 2, 3 and 4. So, all these 3 subunits, all 4 subunits are coming together, and that is how you are going to have the quaternary structures. So, different polypeptide chains are arranged to give the quaternary structure. So, that is why if you want to understand the protein structures, you have to understand all the four different types of structures. So, we have the primary structures, we have the secondary structures, we have the tertiary structures, and we have the quaternary structure. So, these are the different level of organizations what is present into the protein structures. So, let us first start with the primary structures. So, the primary structures, the amino acid sequence of a protein is known as the primary structure. The order of the amino acid determine the folding of the protein to achieve the net minimum free energy, and this is achieved in the multiple steps collectively known as the folding. So, if the primary structure is going to fold, so this is what you see here is the primary structures, right, where you have the length of the amino acids. Now, see here I am just showing you the single letter code of the different amino acid, because it is almost impossible to write the full length or the full name of that particular amino acid, because to save the space, right. And these primary structures are actually going to fold to give you the secondary structures, and that event is called as the folding. So, when the primary structure is going to fold into a proper 3-dimensional conformations, then it is going to give you the secondary structures. Now, the question comes, how we can be able to determine the primary structures. So, if you want to determine the primary structures, you have to first achieve the primary structures, and then you can be able to sequence the protein, and you can be able to know the amino acid sequence of that particular proteins. So, these are the different steps what is being shown. So, what you can have is you can have the starting with the protein structure, right. So, this is the 3-dimensionally folded protein, right. So, here you are going to have the tertiary and secondary structures. Now, what you have to first do is you have to first convert that into a linear chain of amino acid, which means first you have to achieve the primary structures, right. So, it means you are actually going to unfold the protein by the chemical or the enzymatic method. Then what you are going to do is, because this length is going to be very large, then you are actually going to break the peptide or break the protein in small pieces, right. So, into small pieces, and you are going to break the small pieces like this. So, they are going to be overlapping regions, right. So, that you can be able to put them these things separately, and then what you are going to do is you are actually going to label the terminis or the terminal amino acids, right. So, you are going to have the labeling of the terminal amino acid in this way, right. So, you are going to have the labeling. So, labeling with the fluorescent dye in different and then you are going to do the sequencing, right. Then you are going to identify that labeled amino acid. That sequencing you can do by two methods. You can use the Sanger's sequencing method, or you can use the advan degradation method. So, this is what it is showing here, right. First you are going to start with the 3D fold structures, then you are actually going to make it unfold. So, you are going to use the different types of treatments, like you are going to react with the FDNB and all that, so that it is actually going to break the disulfide linkages, and then once it is going to adopt the primary structures, then that you are going to degrade the primary structure with the help of the different types of chemicals or the enzymes, and that is how you are going to get the small fragments. And once you got these small fragments, you can actually be able to do the sequencing, and then once the sequencing is over, then you can be able to put these blocks together, and that sequencing you can do either by the advan degradation method or to the Sanger sequencing method. So let's understand. So in the state 1, this is the stage 1, right? So stage 1, you are going to convert the 3D conformations into the 1D or the primary structures, right? So that you are going to achieve simply by in the stage 1, where you are first going to break the disulfide linkages. So stage one is the breaking of the disulfide bonds. So you can imagine that this is the protein, right, which has a disulfide linkages, and the disulfide linkages interfere with the complete sequencing procedure, as it does not allow the release of the cleaved amino acid from the peptide chain. There are two approaches to disrupt the disulfide linkages in a protein sequence. In the first approach, the protein is oxidized with a performic acid to produce the 2-cystic residue. So why there is a need to break the disulfide linkages? Because if you don't break the disulfide linkages, even if this particular amino acid is actually going to be labeled and it is going to be hydrolyzed, it is not going to be released from the main chain, because it is still having it is bind to the main chain through a disulfide linkages, and that is why it is important to break the disulfide linkages. So disulfide linkages, we have the two approaches. In the approach 1, you can actually use the oxidation with the performic acid, and that's when you do the performic acid treatment, it is actually going to break the linkage between the disulfide linkages, and that's how it is actually going to give you the two fragments. Whereas in the approach number 2, the protein is reduced by the DTT or beta mercaptoethanol to form the 2-cysteine, followed by the treatment with the iodoacetate to form the carboxymethylcysteine. Formation of the carboxymethylcysteine stops the reformation of the disulfide bond. So in the approach 2, what you are going to do is you are going to add the DTT. So DTT is a reducing agent. So once you are going to reduce the disulfide linkages, the SS is actually going to get converted into SH, and that's how you are going to have the two peptide bonds where the disulfide bond is broken. But this has a problem, because as soon as you have the SH and you have the reducing environment, it is going to be remain as SH, but once it is actually going to be acquire the oxidizing environment, again the SS is going to be oxidized, and again the SH is going to be get converted into the SS double bonds. So to avoid that, you are again going to react this with the carboxymethylase by the iodoacetate. So in that case, then what will happen is that the S is actually going to be tagged with this particular functional group. This is going to and that's how it is actually going to form the carboxymethylated cysteine residue. And once you have this, then they will not be able to come together even if the conditions are oxidizing in nature. Now, once this is done, you can actually go back to the stage 2. In the stage 2, you are actually going to break the big polypeptide chain into the multiple fragments. So, in the stage 2 is the cleavage of the polypeptide chain. So, in this stage 2 is the cleavage of the polypeptide chain. The protease and the chemical treatments are targeting protein have a specific recognition sequence, and they cleave after a particular amino acid. So, this is one. So, stage 1 is this, right. So, stage 1 is over, right, where you have actually destroyed the disulfide linkages by the two approaches what we have just discussed, and now in the stage 2 you are actually going to cleave the protein with a enzymatic or the chemical methods. So, some of the common reagents what you are going to use for fragmenting the polypeptide chain is that you can use the enzyme. So, you can, if you use the trypsin enzyme, the trypsin has the cutting site which is actually after the lysine or to the arginine, which means wherever the lysine or the arginine is present. For example, these are the peptides which are being generated by the trypsin. So, if you treat it with the trypsin, it is actually going to cut wherever you have the lysine or the arginine. So, for example, here it has cut, right, here it has cut. So, wherever you have the lysine and arginine, it is actually going to cut, and that is why it is actually going to generate the different types of fragments. Similarly, you can use the chymotrypsin. So, chymotrypsin is actually going to cleave the peptide after the phenylalanine, tryptophan or tyrosine, which means after the aromatic amino acids. So, then we can also use the pepsin. So, pepsin is actually going to cleave the polypeptide chain after the leucine, phenylalanine, tryptophan or the tyrosine. And then you can also have the different types of chemicals. For example, you can use the cyanogen bromide, and cyanogen bromide is actually going to cleave the polypeptide after methionine. For example, these are the peptide sequences what is being generated by the cyanogen bromide. So, you see this is the methionine, and after the methionine it has been cut by the cyanogen bromide. Now, once you got these small fragments, then what you can do is you can take the individual fragments and then you can sequence these small fragments. Once you got the sequence of these small fragments, then you can have to put them together, and that is how you are going to get the sequence of the complete proteins. So, now in the stage 3, you are going to do the sequencing of the polypeptide chain. So, these are the you are going to have multiple polypeptide chains. So, for the sequencing of the polypeptide chain, you can have the two methods. One is you can use the Sanger's method, or you can use the Edmond-Degradant method, right. So, let us first discuss about the Sanger's method. So, once the polypeptide fragments are generated, we can start the sequencing of the each polypeptide chain. It has the following steps. So, first thing is you have to identify the N-terminus residues. The N-terminal amino acid analysis is being performed in the 3 steps. Number 1, you are actually going to label the terminal amino acids. So, as I said, when we were talking about the primary structure, so primary structure has the N-terminal thing and then it has a C-terminal thing, right. So, what we are doing is we are first sequencing the protein from the N-terminus. So, first amino acid we have to first do the sequencing from the N-terminus. So, for the first amino acid we are just using the labeling. So, we are labeling the terminal amino acids. So, the chemical reaction is performed to label the terminal amino acid with the compound such as the Sanger's reagents like 1-fluoro-2,4-dinitrobenzene or DFNB and the densyl chloride. In most of the cases, these reagents also label the free amino acids which are present on the basic amino acid side chain such as lysine and arginine. Dinitrofluorobenzene reacts with the free amino group to form the dinitrophenyl amino acid complex. So, what we are going to do is you are going to take the DFNB and then if you add the DFNB to the first amino acid, because it has the free amino group, it is actually going to, and in the presence of the HF, so there will be a release of this group, and then it is actually going to form a bond with the terminal amino groups, and that is how the first amino acid R1 is actually going to be labeled. Now, the step 2, what you are going to do is in the step 2 you are going to hydrolyze the protein. So, when you add the acid hydrolysis of the dinitrophenyl amino acid complex, that lead to the breaking of the peptide bond to release the dinitrophenyl amino acid complex in the solutions. So, after this, once the first amino acid is being labeled, then you are going to do the acid hydrolysis, and when you do the acid hydrolysis, it is actually going to break the bond between the first amino acid and the second amino acid, and as a result the first amino acid which is already been labeled with the DFNB, it's going to be released from the main chain. Then you are going to do the separation and the analysis of the derived amino acids. So, a HPLC or the TLC separation of the complex and comparing it with the standard amino acid is actually going to give you the name as well as the identity of this. So, what you are going to do is once you got this amino acid, then you can actually be able to run the TLC along with the standard TLC. So, what you can do is, like for example, you can run a TLC like this. So, you can run all the 20 amino acid, right. So, you can actually make all the 20 amino acid in the DFNB complex, and then you can run, and then you can also run the unknown sample, right. So, if you run the unknown sample, suppose it goes the spot here, and suppose this is the arginine, right. So, if it goes to this, then you can say that this is the arginine, or you can actually be able to calculate the RF value of your unknown sample, and since you know the RF value of all the other amino acid complexes, you can be able to identify this. The other approach is that you can do the HPLC and you can be able to calculate the retention values. So, this is about the how you can be able to use the Sanger's method to sequence the proteins. Now, if you talk about the advan degradation method, so in the advan degradation method, it also has the similar kind of steps. For example, the similar to the Sanger reagents, the reagents are different, like where you are actually going to use the phenyl isothiionate reacts with the terminal amino group to form a cyclized phenyl thiocarbomoyl derivative. So, in this case, you are going to use the phenyl isothiocyanate, and when it reacts with the terminal R1 group, right, onto the peptide, then it is actually going to form a cyclized product. Under the acidic conditions, the terminal amino group is actually going to be cleaved from the main chain as a thiazolidine derivative, and that's how you are actually going to have the first amino acid as the PTH. So, thiazolidine derivative is extracted into the organic solvent and it forms the phenyl thiodine amino acid PTH amino acid complex into the presence of acid. So, ultimately you are going to get the PTH complexes. So, this is for the PTH complex of the R1, and then what you are going to do is you are going to run the PTH amino acid complex can be identified by the HPLC or TLC in comparison to the standard amino acid. Now, once you have done this 1 to 4 for the first amino acid, so when you do the first step 1 to 4 for the first amino acid, you can do the 1 to 4 again for the second amino acid, right, because the first amino acid is been released, right. So, the remaining peptide chain is still there, you can use that and again do the another round of this. So, if you continue this like this, it is actually going to keep giving you the amino acid sequence from the N-terminus side. So, if you step 1 to 4 can be repeated for the next amino acid in the polypeptide chain, and that is how it is actually going to give you the whole sequence. And that sequence, if you have the different fragments, if you put them together, it is actually going to give you the complete sequence of all the protein peptides. Now, as I said, the protein is having the two chain types. So, you have the N-terminus side and then you have the C-terminus side, right. So, we have just discussed the method like the Sanger's method or the advan degradation method to identify the N-terminus amino acids, but we can also do the sequencing from the C-terminus, and that is how you can be able to identify the C-terminal residues. So, how we can do that? The C-terminal residues, not many methods are developed for the C-terminal amino acid analysis. The most common method is to treat the protein with the carboxypeptidase to release the C-terminal amino acid and test the solution in a timely dependent manner, right. So, what you can do is you can just treat this with a carboxypeptidase. So, carboxypeptidase is a specific enzyme which actually releases the amino acid from the C-terminal side rather than the N-terminal side. So, if you use the carboxypeptidase, it is actually going to release the amino acid, and these amino acids are the C-terminal amino acids. So, once the amino acid is released, you can identify that amino acid by the Sanger's or the advan degradation methods. Then the stage 4, you are going to do the ordering of the peptide fragments. So, usage of the different peptide cleavage reagents produces the overlapping amino acid stretches, and these stretches can be used to put the whole sequence. For example, when you generate the trypsin, right, it is going to generate the first fragment like this, the second fragment like this, the third fragment like this. So, if you sequence this fragment, if you sequence the A fragment, if you sequence the B fragment, if you sequence the C fragment, so what you see here is that A is having this portion which is overlapping, C is having this portion which is overlapping with the B. So, by doing this overlapping sequencing, you can be able to deduce the final sequence of the final length, or you can be able to put them these fragments, and that is how you can be able to do the sequencing. Then the stage 5, you can actually be able to locate the disulfide bonds. So, the peptide cleaved by the protein cleaved by the trypsin is performed with or without breaking the disulfide linkages. Amino acid sequence analysis of fragment will provide the site of the disulfide bond. The presence of a disulfide bond will reduce the two fragments right, will reduce two fragments and will appear as a single large fragments. As we said, right, if there is a disulfide linkage present, it is not going to allow the release of the amino acid fragments. So because of that, if there is a, suppose this is the disulfide linkages, right, and so even if you cleave this particular fragment, right, so for example, if there is a disulfide linkage like this, right, if there is a disulfide linkage like this, if you cleave this with the protease, it ideally should give you the two fragments, right, but if there is a disulfide linkage, it is still be going to bind, right, this, and that's how it is actually going to give you the single fragment. So if you get the single fragment, then there is a disulfide linkage which is present. Apart from these kind of methods, and with the advancement of the mass data of different types of peptide fragments and all those kind of thing, the mass spectrometry method is also being used. So in the recent past, the mass spectrometry method in conjugation with the proteomics information is also been a popular tool to characterize the each fragment to deduce its amino acids. So in this mass spectrometry data, what you are going to do is, instead of doing this sequencing, what you can do is you can just simply calculate the peptide mass, and that peptide mass actually, because there is a complete database, can be used to deduce the sequence. I have given you the reference. So if you are interested more about reading the protein sequencing, you can actually be able to read through this particular reference. Now let us talk about the secondary structures. So secondary structures, the amino acid interact with each other, and as a result the peptide chain folds into the secondary structures. These secondary structures are the building blocks for the tertiary structures. So these are the primary structures. When they fold, they give you the secondary structures. Secondary structures could be of two types, either it can be alpha helix or to the beta sheets. It is a helical structure termed as the alpha helix by the Linnaeus Pauling. In this structure, the polypeptide backbone is wound around a central axis, with the R group of the amino acid protrudes outward from the helix bond band. In most of the protein, the helix is right-handed, which means you are actually going to see the helix, and in the helix the R groups are protruding outside. So then we have the beta sheets. This is a more extended conformation of the polypeptide chain, where the R groups protrude from the zigzag structure in the opposite direction, giving a alternate structure. So beta sheets could be the two types. It could be the parallel beta sheets or the anti-parallel beta sheets. So it can be parallel or the anti-parallel beta sheets, which means either the beta sheets are running in the same direction, then it is called as the parallel beta sheets. If they are running in the opposite direction, then it is called as the anti-parallel beta sheets. Then we have the turns. So these secondary structures have no definite structures, and they are present in the protein structure to change the direction of the running polypeptide. These are also found to places to connect the successive alpha helix and beta sheets. The number of amino acid and their preference in turn is not consistent. The two protein can adopt the similar 3D conformation by changing the length and keeping the amino acid in the turn region of the structures. So turn is actually a unstructured region, and it is actually not having a defined structure, but it has a very huge significance in terms of providing the flexibility of the different types of protein structures. So for example, you can have the two different types of protein structures, their sequence, their amino acid sequence could be different, but they may adopt the identical structure simply by changing the length of the loops within the length, right. So you can see that these are the loops, right. So what you see here is this green color region is actually called as the loop or the turn. Then we have the tertiary structures. So tertiary structures, secondary structures folds to give the rise the higher order organizations, commonly known as the tertiary structures. And then the tertiary structures can still be packed, and that's how you can have the quaternary structure. So if the multi polypeptides are involved in the constitution of the protein, the tertiary structures of these different polypeptide chain come together to form the quaternary structures. Now as we discuss about the methods to determine the the primary structures, right? We have discussed about the Sanger's method and the advan degradation method. We also would like to discuss about the methods to determine the secondary or the tertiary structures. So, what are the methods to determine the secondary or to the tertiary structures? So, there are two approaches. One is you can use the experimental method. So, experimental methods, there are two methods. So, you can use the X-ray crystallography or the NMR spectroscopy, and these are the two methods which you can use to determine the three-dimensional structure of the proteins. For the X-ray crystallography, you can actually be able to have the very different steps, right? So, what you are going to do is, first you are actually going to isolate the protein for which you are actually you want to identify the secondary or the tertiary structures. Then what you are going to do is you are going to purify this protein at 100% purity or more than 90% purity. And once you are done the purification, then what you are going to do is you are actually going to crystallize this protein. So, you are going to produce the crystals, and once you produce the crystal, then you are actually going to put these crystals for the diffraction. And what you are going to get, so you are going to do the diffraction of these crystals, and once you do the diffraction, it is going to give you the diffraction patterns. So, what is diffraction pattern? Diffraction pattern is actually going to give you the spots around the axis. So, it is going to give you the wherever the diffracted X-ray beam has hit the film, right, and that is how it is actually going to give you the diffraction pattern. And ideally, when you are want to collect the complete diffraction pattern of a protein, it has to be rotated, this crystal has to be rotated for an angle of 360 degree, right, because you can imagine, right, this is the crystal, right, you can have, right, and this crystal actually has to be rotated for 360 degree, then only you can be able to collect the diffraction pattern of or diffraction of all the electrons what is present. Once you have the diffraction pattern or the diffraction data, right, from the X-ray, then you are actually going to put this and you are going to analyze the diffraction data, and that is how you are actually going to get the electron density map, right. Once you collect the electron density map, it is actually going to give you the position of the electrons within the three dimensionals, right. So, once you got the electron density map, then you are actually going to fit the protein molecules. So, you can to fit the protein molecules, molecule, right, protein, which means the protein sequence, you are going to fit the protein sequence, and once you are done the fitting, then it is actually going to give you the 3D structure of the protein. Once you got the 3D structure of the protein, then you can actually do the quality assessment, right. You can do the quality assessment with the help of the 3 programs. You can do the Ramachandran plot, you can do the ProCheck, and you can also do the errata plot. So, if you do all these kind of thing, it is actually going to tell you whether the 3D structure what you have solved by fitting the protein sequence into the electron density map is correct or not. If you get the R factor, which is called as the error factor, which is approximately around 20, then you are going to say that the protein what you have solved or the protein structure what you have solved using the X-ray crystallography is very good. If you want to read more about, since this course is not about the X-ray crystallography, you will find a very good that kind of course on even in the MOOCs, or as well as you can actually get a lot of good resources if you are interested to understand each and every detail about the X-ray crystallography. You can actually be able to even go through with this particular article, and that actually will give you the very good idea about the X-ray crystallography as well as the NMR spectroscopy. Same to this, NMR also NMR spectroscopy also has the different types of steps where you are first going to use the protein. You are going to the first step is you are going to purify the protein, right, and you know that the normal protein what we are going to get from the bacteria is not going to be labeled, not going to be NMR sensitive, because it's so first thing is you are actually going to label the protein with the NMR sensitive nuclei, right? For example, you can use the N14 nitrogens, you can use the carbon 14, and you can also use the hydrogen like the deuterium and so on. And because of that, the purified protein what you are going to produce is going to be NMR sensitive, and then you are actually going to collect the NMR data. And once you are going to collect the NMR data and you are going to analyze that data, it is actually going to give you the protein structures, and that protein structure is actually going to be called as the average structure, right? So this protein structure is called as average structure because the NMR is actually going to be performed in the liquid. So you can imagine that if I have a protein into the test tube, so this protein actually is freely moving, right? So, and its domain and all other kind of structures are also moving. Because of that, it is actually going to give me the average structures. Apart from that, you can also have the non-experimental methods, such as you can also do the homology modeling. So this is a useful and a fast structural solution method where the sequence similarity between the template and the target enzyme is used to model the 3D structure of the target enzyme. The homology modeling exploits the idea that the amino acid sequence of a protein direct the folding of a molecule to adopt the suitable three-dimensional conformation with the minimum energy. So what you are going to do is you are going to take, so in this homology modeling depends on the reliability of or depend on the phenomena that the two proteins, when they are actually having the similar kind of amino acids, they are actually going to adopt the similar type of folds, right? Because you know that the primary structure is actually going to direct the folding of these amino acids, and that's why when the primary structures are when the primary structure or the amino acid sequence is identical, it is actually going to fold into the same cell, which means if you want to use the homology model, you are actually going to have the two things. You are going to have a template structure, right? So you're going to have a template structure, and you are also going to have the test amino acid sequence, right? Now, first what you have to do is you are actually going to use this particular template, and you are going to use this sequence, and you are going to do the multiple sequence alignment. So you are going to do the sequence alignment, and this sequence alignment is actually going to tell you whether this particular template is good for modeling this particular amino acid sequence or not. Once this is done, then you are actually going to do two things. You are going to take the structure information from the template, right? And on this structure information, you are actually going to put the amino acid sequence what you have from the test, and that's how you are actually going to prepare the modeled protein. So once you got the model protein, then what you're going to do is you are going to test the quality of the model. And we have already discussed the quality of the test of the model can be done by Ramchandran plot, or you can use the errata plot, or you can do the ProCheck, or you can do the verify 3D. So these are the 3 or 4 different types of groups or the different types of programs what you can use, and you can be able to do the error measurements, right? Once you have found that the structure is good, then what you can do is you can actually be able to utilize this model for the different types of applications. The programs what you can use very oftenly is called as the modeler. So you can already use the modeler 9th version, and that is actually going to allow you to do the modeling. So all these steps you can actually be used with the help of the modeler, and then you can use the different types of programs to do the to do the quality of the model verification with the Ramchandran plot and all these kind of things. So now let's move on to the methods to determine the quaternary structures. So method to determine the quaternary structures. You know that the condition for the quaternary structure is that the protein should have the multiple subunits, right? So if the protein has the multiple subunits, then only you can be able to perform the quaternary structures. So how we can actually be able to know that the protein has the multiple subunits? So what you can do is you can calculate the protein's molecular weight under the two conditions. One, you can actually be able to calculate the protein's molecular weight under the native conditions, or you can actually be able to calculate the protein's molecular weight under the denaturating conditions. So when you denaturating conditions, imagine that the molecular weight under the native condition is mole n, so it is n, right? And under the denaturating conditions, if the molecular weight is mole d, then the you can actually be able to calculate the oligomeric status by the molecular weight native divided by molecular weight denatured, right? For example, so like molecule n versus molecule d. Let's take an example. Okay, for example, if I have calculated a molecular weight, and if I calculated the native molecular weight is 120 kDa, and if I have calculated the denatured molecular weight, which is the 30 kDa, then the oligomeric status would be the 120 divided by 30, which means the 4, which means it's a tetramer. So once I calculated that it is actually a tetramer, then I can be able to assure that there is a quaternary structure what is present. Now the question comes, how you can be able to calculate the molecular weight of a protein under the native or the denaturating conditions. So calculation of molecular weight, right? So for the native molecular weight, what you can do is you can run the protein under the gel filtration chromatography. So if you do the gel filtration chromatography, uh, although the scope doesn't allow you to explain the gel filtration chromatography, but gel filtration chromatography is a chromatography technique which is actually going to filter the molecule based on the size. So if it's based on the size, so if it is going to be 4 times, because you are taking the one monomer and you are making the 4 monomers, the size is going to be 4 times, right? So that's why it is actually going to give you a pattern, right? It's going to give you a peak which is actually going to tell you that okay, this is the size at which the protein is eluted. So this is called as the elution volume, and utilizing this information and as well as the distribution coefficient, you can be able to calculate the molecular weight of this particular protein, and that is going to be called as native molecular weight. And now under how to calculate the denatured molecular weight, you can be able to calculate the denatured molecular weight with the help of the SDS page. I'm sure we all know about the SDS page. So if you run the protein on the SDS page, it is actually going to give you the molecular weight, right? So if you run, first you run the molecular weight marker, right? So you run the marker, and then you're going to run your proteins. So depending on the, and then you calculate the RF values for the these marker proteins, right? So if for the individual marker protein, when you calculate, you are actually going to get the RF values for each and every spot, and then you can also be able to calculate the RF value for the your protein, and depending on the RF value of this particular spot, you can be able to calculate the denatured molecular weight. I have already discussed this whole thing in a and so that's how you can be able to calculate the denatured molecular weight. And once you have the native molecular weight and you can have the denatured molecular weight, you can be able to utilize that for calculating the oligomeric status, um, which is the molecular weight native versus molecular weight denatured, right? So, with this we have discussed about the protein structures. What we have discussed about the different types of organization, what is there in the protein structures. We have discussed about the primary structures, we have discussed about the secondary structures, we have discussed about the tertiary structures, and we have discussed about the quaternary structures. We what we have so with this we have discussed about the protein structures, and what we have discussed so far, we have discussed about the primary structures, secondary structure, tertiary structure, and quaternary structures. While we were discussing about the primary structures, we have also discussed about the method to determine the primary structures. So, we have discussed about the Sanger's method or the advan degradation method. So, both the methods are utilizing the similar approach, where you are actually going to label the terminal amino acids, and then you are going to use the acid hydrolysis, so that the terminal amino acid is going to be released, and that terminal amino acid, the labeled terminal acid, going to be identify by running it onto the thin layer chromatography. And apart from that, we have also discussed about the secondary as well as the tertiary structures. We have discussed about the methods to determine the secondary as well as the tertiary structures. And then lastly, we have also discussed about the homology modeling. We discussed about the X-ray as well as the NMR spectroscopy. And lastly, we have also discussed about how you can be able to determine whether the protein is going to have the quaternary structures or not. So with this, I would like to conclude my lecture here. In our subsequent lecture, we are going to discuss some more aspects related to proteins. So with this, I would like to conclude my lecture here. Thank you.